In chapter 3, we're going to be dealing with Mendelian genetics, and this focuses primarily on the foundational understanding of patterns of inheritance that stem from the original work of Gregor Mendel. This is a topic that you've certainly covered in introductory biology and likely have had before you came to college, perhaps as early as middle school. And from that work, these Punnett squares probably look somewhat familiar. So we're going to dig into how this research by this gentleman, Gregor Mendel, an Augustinian monk, laid the groundwork for our present understanding of patterns of inheritance of genetically controlled traits. And this is the basis for all of what we're going to be discussing for the rest of the semester. So this fellow Gregor Mendel was an Augustinian monk, as I said, and worked in a monastery, and I guess there wasn't a great deal to do because not only did he grow plants, pea plants for food, he became intrigued by the remarkable variety of pea plants available to him. So plants that were tall or short, plants that had pink flowers or plants that had white flowers. All of these variations intrigued him, and in his work he began crossing different plants. So just as you could get two different labs and breed them together to get puppies, you can actually do this with plants. It's a little different process, of course, but he was able to do this and crossed tall plants with short plants, pink flowered plants with white flowered plants. And the results and his understanding of what was going on and the documents that he wrote up detailing his findings are the basis for genetics as we know it today. And so in this, he studied not only flower color, as we'll get into in a moment, and other traits, but he determined that these traits are controlled by what we now call a gene that occurs as a pair, one on each chromosome. So he didn't necessarily know that chromosomes existed, but he knew that each plant had a pair of copies of information that determined what color its flowers would be. Okay, so this fella had no knowledge of chromosomes or the process of meiosis that you've just learned about in chapter 2. He, in this work, determined what he called units of inheritance exist. Again, we call these genes. He was able to predict the behavior of these units. So he was able to predict when he crossed two plants, say a tall and a short plant, exactly what the resulting offspring would look like based on this understanding that he had developed. So with these pea plants, which to him were a model organism, and a model organism is a term that you're going to hear pretty frequently throughout this semester. Fruit flies are a model organism. Lab mice are a model organism. They're model organisms because they're relatively easy to keep and rear and illustrate principles that are applicable to organisms that are more difficult to keep in the lab. So the pea plants for him were a model organism, and in following these 7 traits, he determined that each of the traits for which there were 2 forms, so tall plants, short plants, pink flowers, white flowers, he was able to secure true breeding forms, true breeding lines. So a plant that, if you bred it to itself, it always produced tall plants. Or a plant that if you bred it to itself, its flowers were white, all of its offspring's flowers were always white. So he had these lines that were very pure for these particular traits, but then what he did was mix them and observe the results. So this is like you perhaps getting a yellow lab and a black lab and breeding them together and observing the results. Who knows what's going to happen, but the interpretation of the results and the continuation of his experiments through multiple generations are where his truly important contributions to the field of genetics lie. And so the 7 traits that Mendel considered are listed here: flower color, position, seed color and shape, pod color and shape, and the height of the plant or stem length. And you can see that there are only 2 different traits. Now, this is a very simple system. It's very simple in that there are only two different character states or phenotypes for each trait. So for seed color, there's only yellow or green. Now, you can think of many other traits that are variable, say even within humans. Think of eye color. There are not simply two eye colors. Even the color of labs. There's chocolates, there's yellow labs, and there are black labs. Okay? He picked a very simple system, and it's in part because of the simplicity of this system that he was able to make the inferences that he was. And so he started out with the simplest possible experiments, what we call monohybrid crosses. So these are crosses of two individual plants that differ only in a single trait. Notice that these two plants differ only in their height, not in the color of their pods, nor their flowers, or any other traits. Okay? So we're going to work through monohybrid crosses first, and then we'll get into more complex scenarios later. So here you can see an illustration of a cross, and all we're showing are the affected characters. And in this case, that is the color of the peas. So we'll just assume the plants are the same height, the flowers are the same color, the pea pods are the same shape and color, but all we're interested in right now are the color of the peas. And if you cross a green pea plant and a yellow pea plant, okay? So we're looking at two generations of this cross, and this becomes important as you'll see in a moment. But first I'm going to get some terminology out of the way for us. And the terminology I want you to become familiar with are F1 and F2, okay? Now, the top two pea pods you see here represent two different parents, a parent with green peas and a parent with yellow peas. They are on the same level or horizontal plane, and they were crossed with one another to produce a first generation of offspring, the F1s. So this first filial generation, okay? So filial is a Latin term that refers to siblings or offspring. And so this first filial generation is produced by crossing the parental generation. And then, as you might imagine, this third row that we see represents the second generation. So the second filial generation, or the F2s. Now, make sure you understand that the F2s are produced not by breeding the parents a second time. It's you breed the parents to get the F1s, and then you breed the F1s to get the F2s. Okay? Now, with pea plants, this is a very simple and elegant system in which to do this, because you can use a single plant from the F1 and breed it to itself. So the plants they produce both gametes, and so they can self-fertilize. And so that's a good example of an F1 cross. You're crossing an F1 individual to an F1 individual and producing F2 offspring. Okay. You could also cross siblings from the F1 to get an F2 generation. Okay, so let's look at the phenotype or the appearance of this F1 generation. All we see are two individual pea pods, and they are both yellow. Okay. Now, Mendel observed this when he crossed his true breeding pea plants to one another when he did these monohybrid crosses and crossed them to individuals that were also true breeding but differed in character state. So crossed a green pea plant to a yellow pea plant, or a tall to a short. Every time he would get an F1 generation, didn't matter how many offspring he had, all of those offspring had the exact same phenotype. They were all the same. So it's as if he crossed yellow labs with black labs and got all black labs. Okay? That's what we see illustrated here. And these two individuals that are illustrated in the F1s are reflective of all F1 individuals. Okay? Now, this clued Mendel into the fact that some of these character states or traits, trait values, are dominant to the other. Okay, so in this case, the yellow color of the peas, the expression of that yellow coloration, is dominant or dominates the expression of green coloration. Doesn't matter who the parents are, like what the biological sex is of either of the parental plants, whether the male plant is the green peed plant and the female is yellow or vice versa. Always, always, always the F1s shared the same character state, and it was only that of one of the parents. So when the F1s are allowed to self-fertilize, producing an F2 generation, you can see that the green peas show up again. Okay, so this character state or phenotype of having green peas, which we now know to be, using current terminology, recessive to the yellow coloration of peas, this shows up again in the F2 generation. Okay, when you have a monohybrid cross like this, and one of the traits, one of the phenotypes is recessive to the other, you will never see it in the F1 generation. And that will make sense when we work through a Punnett square here in a few moments. Okay, so let's look at the results of Mendel's crosses of all 7 of these traits in pea plants. And you can see every time he crossed one of these to another, in the F2 generation, okay, there's no indication of the frequency of the different offspring in the F1, because all of the F1s are going to be what? So all the F1s of the purple flower plant crossed to a white flower plant are going to have what color flowers? They're all going to have purple flowers. All monohybrid crosses in which one trait is dominant to the other will produce F1s that are 100% exhibiting that dominant trait. Okay, so you can imagine for all 7 of these crosses what the F1 offspring phenotypes were. So for flower color, they were all purple. For position, all the flowers were axial. All the seeds were yellow and round, and the pods were inflated and green, and the plants were tall. Okay? Now, what's important, and the only numbers that are presented here, are the frequencies of individuals in the offspring, the F2 generation offspring, that exhibit purple or white, yellow or green peas, tall or dwarf height. And what you will see is that the numbers of individuals exhibiting the dominant trait, say purple, is much higher than the number of F2 offspring that exhibit the recessive trait. So for purple, in the F2, the purple-white cross, in the F2 generation, there were 705 plants that Mendel observed had purple flowers and 224 that had white flowers. Okay? This comes down to a 3.15 to 1 ratio. For every 1 white-flowered plant in the F2, he would get about 3.15 purple-flowered plants. And look down that far right column at that ratio. For almost every trait, the ratio is just about 3 to 1. No matter how many offspring he had, you can see that there are many more F2 offspring for some of these crosses, for example, the seeds, than there were for others. But the ratio is almost always roughly 3 to 1, okay? And if you think back to the previous slide in that F2 generation, remember there was 1 pea pod that had green peas and the other 3 had yellow. And that ratio was intentional for that illustration. You're going to get about 1 green pea plant for every 3 yellow pea plant, okay? And so this ratio is just the all-important aspect of these monohybrid crosses for the F2 generation. Again, remember, these are the F2s. Now, one important thing that I've mentioned briefly is that Mendel conducted reciprocal crosses, in that he crossed plants that had green peas with yellow peas, but he did that not only with male green pea plants and female yellow pea plants, as you see on the left, he also switched it up and used female green pea plants and male yellow pea plants. Okay? The important thing that he observed was that the results were identical. No matter what the gender of the green pea parent was, the cross results, the F1 and the F2s, had the same frequencies of green and yellow peed offspring. So, from this, he proposed this existence of what he called unit factors that pass unchanged from one generation to the next, and they determine the expression of the appearance or the phenotype of this particular trait. We now call these genes, but this is a term that's come into use long after Mendel's passing. So he proposed a few things. The first was that these genes occur in pairs, and this took some considerable thinking. There was a lot going on to reach this conclusion, okay? And we'll dig into how these things occur in pairs within individuals. His conclusion of this relationship between the copies of genes that had would result in different phenotypic traits, so say color or height, he determined that some of these were more dominant than others. They were more likely to be visible and apparent in the offspring than others, depending on the circumstances. So this inference of dominance and recessiveness dates back to Mendel's work on pea plants. And then he also proposed this all-important concept of genetic segregation, okay? And this has everything to do with when gametes are produced, these unit factors, or what we now know to be genes, they separate and go into the gametes. Now remember, the first postulate here is that these genes occur in pairs, and what he's saying is when these pea plants are producing their gametes, their sperm and their egg, that pair of genes separates and only one of them goes into the gametes. This is an incredible inference, but completely jives with what you just learned when we talked about meiosis in chapter 2. And I'll bring it back to meiosis here shortly. Okay, so let's look at a simple Punnett square of an F1 and then F2, so a monohybrid cross of plants that differ in their phenotype. So here we have, in the parental generation, a plant with purple flowers crossed with a plant with white flowers. Now underneath them are letters indicating the copies of the gene that they possess. And remember, in these monohybrid crosses, the parents are true breeding, okay? And there's a term that we use for that, but in this case, it means that the purple flowered plant has 2 copies of the gene that determine flower color. Both of them are the same, and they both produce purple coloration. The white flowered plant has 2 copies of the gene that determines the color of flowers. Both of them are the same, and both of them code for white flowers. And so the 2 letters that occur under each of those parental plants or their flowers indicate the 2 gene copies that each parent has. Now think about what's happening when they produce offspring. That purple flowered plant is going to give one of those copies of that flower color gene, denoted by a large R, to its offspring. And the white flowered plant is also going to give one of its copies of the gene, denoted by a small r, to its offspring. So the F1 generation receives a big R from the purple flowered parent and a little r from the white flowered parent, and they therefore possess one of each, but they exhibit only the purple coloration. And that's because purple is dominant to white. Now, this inside this red box is what we call a Punnett square, certainly something that you've seen before, and this illustrates the crossing of an F1 parent to itself or an F1 parent to another F1 individual, and the 4 individuals illustrated within the box indicate the 4 potential outcomes of fusing an egg and a sperm from one of these F1 individuals. So let's look first at the horizontal top 2 rows, so the pollen, and there's a big R and a little r. Basically, imagine an F1 parent that is going to give one copy of that R gene to its offspring. Okay? Each parent contributes one copy of the R gene, and so the offspring will then have 2. They get 1 from each parent. Now, what R could be put into the pollen? Well, it could be the big R, the left column, or the small r, the right column. Okay? Now, that pollen is going to fuse with an ovule or an egg, and what could an F1 individual put into that ovule? Well, it could be either a big R or a little r, so those 2 rows. Okay? So if you look, each of these squares in the grid in which there is a flower has underneath that flower 2 letter R's. And they are essentially the fusion, the combination of the R at the top of that column and the R at the left of the row. Okay? So the top left flower has 2 big R's, the bottom right flower has 2 little r's. Okay? And the bottom left and top right corners have 1 of each. And it's simply because the first column gets a big R from the pollen, the second column gets a little r from the pollen, right? You see where I'm going here, okay? So these are the 4 possible outcomes, okay? And I want you to think about this Punnett square for a minute because we do a lot with these. And as you look at that, is there any reason that you would think that of all the pollen that these F1s create, more of those pollen have a big R than a little r? Think about how meiosis works. Think about that plant essentially making sperm, okay? Well, that sperm makes what, 4 sperm, or sorry, that plant makes 4 sperm at a time, right? For each meiotic division, 1 cell becomes 4 sperm. How many of those sperm have a big R? 2. And how many have a little r? 2. Okay? So think of all the sperm or pollen that plant makes. What proportion of them have a big R? 50%. And 50% have a little r. The same is true of ovules, right? Despite the fact that oogenesis results only in a single mature ovule, right? Half of those are randomly going to have a big R and half of them are going to have a little r. And so as you think about what this Punnett square represents, recognize that the chance of getting a big R from the pollen and a big R from the ovule, right? It's a 50% chance you get the big R from the pollen. There's a 50% chance you get a big R from the ovule, okay? It's the same as the chance of getting a little r from the pollen and a little r from the ovule, okay? So this square is not only illustrating all the possible genetic combinations, it's also giving you some indication of how frequent they will occur, okay? So we're going to talk about that quite a bit more here shortly. But recognize that the Punnett square illustrates a bit more than you've probably recognized to this point. Okay, so let's cover some terminology, some of which I've already used, but I want to make sure you're quite comfortable with it. And the first is phenotype. And in general, we think of phenotype as being the appearance. Okay, so your Labrador retriever might have black hair, so it's a black lab. That is its phenotype. Okay, I'm tall. I have a beard. Those are aspects of my phenotype. Okay, so your eye color is an aspect of your phenotype. Now, in genetics, we're talking primarily about, almost all the time, phenotypic characteristics that are controlled by genetic mechanisms. Okay, so your eye color is not determined by how often you're outside or how much sun you get, right? It's something that's genetically determined, you inherited from your parents. And so it's reasonably safe for you to think of phenotype as the appearance of an organism. Okay, so your phenotype might be tall or short. It might be that your hair is black or brown. Those are aspects of your phenotype. Now, I put an asterisk after appearance because I want you to understand, it's really important that you understand that not all phenotypic traits are visible. You can't actually see them, right? And so there are many things that are genetically controlled. You inherit them from your parents, but I can't see them when I look at you, okay? They are part of your phenotype, but it's not visible, okay? So your blood type is a great example, okay? Your blood type is a real thing. It could cause serious problems if it's ignored in a medical setting, right? You should be aware of that. But I can't see it. It's a genetically controlled phenotypic trait that you can't see, okay? Your sense of smell, right? Perhaps you have this very strong negative reaction to some odors, right? That could be genetically controlled and not something that I can see when I look at you. An interesting phenotypic trait that is genetically controlled, and they're now telling you about when you get some of these consumer genetic testing things done, like 23andMe, is if you eat asparagus, does your pee smell funny or strange to you? So if you've eaten asparagus before and then urinated and noticed a funny smell, that means you are able to smell that, okay? It doesn't mean that the people that don't smell it, their pee doesn't smell different. It's simply that they can't smell it. Their phenotype is that they are incapable of detecting that odor, okay? So that's a phenotypic trait that we can't see. So just keep that in mind when we use the term phenotype. All right, now gene, that's a term that you should be comfortable with. These are the unit factors that Mendel knew about and understood. They come in pairs in organisms like ourselves that are diploid. But this term is often confused with alleles, okay? And it's important that you understand the term alleles refers explicitly to different forms of a particular gene, okay? So the way I tend to explain this is thinking of a gene as a recipe, okay? Now, you and I might have a recipe that we use for chocolate chip cookies, and when we execute that recipe, we both end up with chocolate chip cookies. But my recipe is different from yours. The end product is a chocolate chip cookie, but they're not identical. The same is true of genes. I have brown eyes, and if you have a different colored eye, or two eyes, hopefully, if your eyes are different colored than mine, it's because we have the same gene that makes some sort of pigment in our eye, but we have different versions of it, just like you and I have different versions of the recipe for chocolate chip cookies, all right? So the alleles that you possess, the combination of alleles, are what determines your phenotype. If you're a pea plant and you have two big R's, two purple alleles, then your flowers are going to be purple, okay? It's the R gene. The allele is just whether it's a capital R or a little r, okay? If you have a big R and a little r, you have two different alleles for that flower colored gene, all right? Now, This is an important distinction, and I will be consistent with it in my discussions and lectures and in my questions on exams. But I want you to understand this because this is a very important distinction. And from this point on, I expect you to notice when you're listening to news or watching movies, or you hear people talk about some genetic test, okay, you will recognize that when someone says, oh, she got tested and she has the gene that predisposes her to breast cancer, or she has the gene for cervical cancer, that's horseshit. Okay? We all have the same genes. Okay? What we have that are different are alleles. We all have the gene, but our alleles are different and result in different phenotypes. Okay. So, how do we denote these alleles? And another term that we're going to be using is genotype. So your genotype is basically what two versions of the gene do you have? You have two copies. What are your two alleles that you possess? Okay. Now, these alleles are generally indicated by one or a couple letters sometimes. But usually for our purposes in this class, we will use a single letter. An allele would be something like the big R or the little r from that flower example. The genotype of an individual is the combination of the two copies of that gene that the individual received from both of their parents, one from each. And what is that combination? Is it big R, is it big R, or is it little r? Here you can see that this genotype I've illustrated is two little d's, all right? Gene names are generally, or these alleles and gene names are generally italicized, and the allelic indicator, like this D, is usually italicized, and it's an abbreviation of some name for the gene, okay? So the gene might be dwarf, right? And if you have the recessive allele, you're a short plant. Okay, so generally the dominant allele is capitalized and the recessive allele is lowercase. This is not always the case. There are other situations where there isn't a dominant or a recessive. Things get a bit more complicated, and we'll be digging into that a bit later. So don't worry too much about that just now. Four terms really that I want you to be very comfortable with are homozygous, heterozygous, homozygote, heterozygote, okay? And individuals are homozygous, it's an adjective, it's a state, if they have two identical alleles, if they have two little d's, or if they have two big D's, they are homozygous. Homo means same, okay? And so homozygous individuals have two copies of the same thing. The term homozygote refers to an individual that has two of the same thing, okay? So I might say, I am homozygous for this allele, or I might say, I'm a homozygote, okay? Heterozygous refers to individuals that possess two different alleles. So in this instance, a big D and a little d, okay? An individual that is heterozygous is called a heterozygote, okay? Now, you have probably learned to use this first term, homozygous, in combination with the term dominant or recessive. So in these two examples here, you'll see two little d's, and that individual we would say is homozygous recessive. An individual with two big D's would be said to be homozygous dominant. Now, we don't use either of those with heterozygous. Okay, I frequently have students that will say they're heterozygous dominant, or this individual's heterozygous recessive. You don't use dominant or recessive with heterozygous because there's just one of each. Okay, you've got a big D and a little d, you're heterozygous. There's no need to put dominant or recessive after that. It doesn't add any information. If you think about that, that ought to make sense to you. Okay. So let's again talk about these Punnett squares and how these can be used to predict the outcomes of genetic crosses between individuals. Okay? And so basically what we're doing is on paper creating all possible offspring for a particular cross. Okay? So each possible gamete is assigned a column or a row. Now the textbook uses by convention all the columns are the female gametes and the rows are the male gametes. It really doesn't matter. You could switch them up and you're going to get the same thing. It won't change what's in the Punnett square, the contents of the square. Okay? So let's look at a Punnett square here where we cross a tall pea plant with a short pea plant. Okay? And so you can see on the top, we've got the symbol for female. So the two columns are going to be the alleles that the female is providing to the offspring, and the male symbol on the left, the male is short. A true breeding short plant will be homozygous recessive, of course. So let's look here. This tall true breeding female, homozygous for the big D allele, is crossed to a true breeding short male that is therefore homozygous recessive. So we've got a homozygous dominant female crossed to a homozygous recessive male. And you can fill in these blanks simply by basically adding the row and the column together. And you'll see that in this F1 cross, all of the offspring are going to be tall. But what is their genotype? They all have a big D, they all have a little d, and they are heterozygous. Okay? And for this reason, we don't talk a lot about the F1 offspring of a monohybrid cross. Because what is the height of all of these offspring? They're all going to be tall. They're all heterozygous, they're all tall. All of the same genotype, all of the same phenotype. They have the same appearance and genetic combination of alleles. Now it's the F2s that are going to be interesting. So what we have here is the female and the male are both tall because they are these F1 offspring. So they have a big D and a little d, each of them. And they are going to contribute equally to their offspring. Half of their offspring will get a big D, half of their offspring will get a little d. And therefore, the combinations that are created in the offspring are going to be as illustrated here in this Punnett square. We're going to have, for every 4 offspring created, I expect that one of them will be homozygous dominant. I expect that one of them will be homozygous recessive. And then 2 of them will be heterozygous. They will have a big D and a little d. Now note that those 2 heterozygous outcomes are heterozygous for different reasons. The top right one got the recessive D from the mother and the dominant D from the father. If you look at the bottom left heterozygote, you'll see that it's completely the opposite. They got the dominant D from the mother and the recessive D from the father. Okay? So there are 2 ways that heterozygotes are created here in an F2 monohybrid cross. All right. So the F2 offspring are mixed. They are mixed not only in their genotype. As you can see, there are lots of different genotypes here. There are actually 3. There's homozygous dominant, homozygous recessive, and heterozygous. Okay? But there are also 2 different phenotypes in these offspring. Look at these 4 offspring, possible offspring outcomes. How many of them are tall? That would be 3 out of the 4. The only one that is short is that homozygous recessive individual on the bottom right. So what we have in the results of the F2 cross is a genotypic ratio of 1 to 2 to 1. For every 1 homozygous dominant individual, you get 2 heterozygous individuals and 1 homozygous recessive individual. Okay? 1 to 2 to 1. Okay? Now the phenotypic ratio is quite different. For every 3 tall plants, we expect to get one short plant. Now, the power of this Punnett square is that it not only shows you all the possible combinations, but because each of these columns and rows are each equally probable in the creation of sperm and egg, then each of these genotypes in the F2 offspring are equally likely to happen. So each of these 4 squares is equally likely. If this plant were to have only one offspring, there would be a 25% chance that it is homozygous dominant, a 50% chance that that one offspring is heterozygous, and a 25% chance that the offspring is homozygous recessive. So these Punnett squares are not just showing the possible outcomes, they're also showing their relative frequencies. So if there are 400 offspring produced from this pea plant cross, how many of those 400 do you expect to be short? Well, the only short ones are the homozygous recessive individuals. There's one of those for every 3 tall individual, okay? It's a 3-to-1 ratio, tall to short. For every 4 individuals, there's one short one. For every 400, there should be 100 short individuals, okay? So these ratios and the possible combinations are much more powerful than you might have understood to this point. So here we've got 2 genotypes that have the same phenotype. And this is an important consideration for you as you interpret Punnett squares. All 3 of these individual allelic combinations are going to be tall plants, okay? So unlike the homozygous recessive individual, which has a very different phenotype, it's a short plant, 3 out of 4 of our offspring are going to be tall. But I know that among those 3/4, that some of them are homozygous for the tall allele and some of them are heterozygous. So how can we determine the genotype of a tall individual? Now, I could, in my lab, sequence this gene. I could sequence their genomes and tell you whether an individual is homozygous or heterozygous, but Mendel didn't have that. And he used his ingenuity in order to cross more plants and test through what we call a test cross what the genotype of individuals that exhibited this dominant phenotype actually was. And so in a test cross, we cross an unknown genotyped individual that possesses or exhibits the dominant phenotype. We cross them with a recessive homozygote. Now I want you to think about this for a moment. This recessive homozygote, that plant is short. If you're Gregor Mendel and you know that this short allele is recessive, the only way an individual plant could be short is if it is homozygous for this recessive allele. So he knows that that short plant, he knows what its genotype is. For the tall plant, he never knows, right? Unless it's an F1 where he knows it's heterozygous. But if it's a tall plant resulting from an F2 cross, how could he possibly know the genotype? All he knows is that they have one copy of the dominant allele. That second copy of the gene could either be another dominant copy or it could be a recessive copy. Let's look at why his test cross was such an ingenious mechanism for him to infer the genotype of these unknown dominant phenotyped individuals. So if you take this tall individual for which you are not certain of its genotype, you know only that it has a dominant copy of the gene, this big D allele in the first column, and you cross it to this recessive homozygote, this short plant, so you know the only alleles that that short plant is able to give to its offspring are the recessive D, okay? Think about what's going to happen here. There are 2 possible genotypes for that tall plant. It could be homozygous for D, the big D, dominant D, or it could be heterozygous. So, if this unknown tall individual is homozygous, meaning they have two copies of this large D, then what combination of alleles are we going to have in this far right column? Here you can see that we will end up creating 2 more out of 4 heterozygotes. So all of these offspring are going to be tall. All of them will be heterozygous. So if in this test cross you cross this unknown tall individual with a short homozygote, a recessive homozygote, all of the offspring are going to be tall. So if all the offspring are tall, you know that that individual, that tall unknown individual, now you can say conclusively that they were homozygous for that dominant allele. If, on the other hand, that tall individual is heterozygous, then we have another possible outcome in which that far right column, when you fuse the row and column alleles together, we get homozygous recessive individuals. Okay, so half of the offspring would be homozygous recessive, therefore short. Half of the offspring, the left column, would be heterozygous and tall. And so in a test cross, we're going to see half of the offspring being tall and half of the offspring short. So here's a real life example that I actually ran into. So I'm a herpetologist at heart, meaning I study reptiles and amphibians, and I've loved them since I was a child and always had lots of pet reptiles. So snakes, lizards, turtles, also had lots of frogs. And I've had a few boa constrictors in my life, and this is a particularly unattractive boa constrictor that for the most part you should be able to buy quite inexpensively. So this is a juvenile ugly boa constrictor that I reckon you could probably get for about $75. But when I was looking at some online classifieds for snakes in order to create an example like this, I found the following listing of a similarly ugly boa constrictor for not $75, but this gentleman was asking $450 for this boring looking boa constrictor. Okay. Now, there's all sorts of stuff here. It tells you, it's a healthy snake and it eats. He's also a biter, it seems, which is always a top selling point for snakes. But what I want you to pay attention here is, okay, why would anybody pay $450 for what looks to be a $75 snake? Well, what this seller is telling you is that you're paying $450 for this snake because there's a 66% chance or possibility that this individual is heterozygous for this T plus tyrosine positive albino. Well, what the hell does that mean? Well, you understand what a 66% chance is, right? Now remember, if this snake is heterozygous for something, if it carries some recessive allele, what it's exhibiting is the dominant allele, the normal, boring, ugly color. An albino boa constrictor looks like this, okay? And these sell not for $75, but at the time, about $4,500, okay? So now you might understand why somebody would pay $450 for a boa constrictor that's heterozygous for this. They carry the albino allele. They look brown and boring because they have the dominant allele. But if they have that recessive allele, what could you do? You could cross two individuals. Here's a cross. Two heterozygous individuals, okay? The parent up top and the parent on the left are both heterozygous. Both of those individuals would be boring brown boa constrictors, okay? But they both carry that recessive allele. Now think about what happens when you cross the two of them. I might pay more money for a snake that's heterozygous if I can cross it with another heterozygote and look at the offspring here. Are all of those offspring going to be boring brown boa constrictors? No. That bottom right, the homozygous recessive individual, 1 out of 4 offspring are going to be albino. Okay? That homozygous recessive condition, those are the individuals that are worth a great deal of money to snake breeders. Okay? So now you might understand why I might pay more for a snake that is heterozygous. Okay? So how could I get a snake that's heterozygous? Well, that's pretty easy. You could do a monohybrid cross, breed an albino snake, cross it with a normal homozygous dominant snake, and all the offspring, all the F1s would be heterozygous for albino, right? Just like those pea plant crosses that we did before. But what this individual said in the advertisement is that the snake he's selling for $450 has a 66% chance of being heterozygous. Well, how the hell does he know that? Okay, well, if you look at this cross, I think you can figure it out. If he crossed an albino and a wild type, brown snake that's homozygous dominant, all of his offspring would be heterozygous. Okay, and a true heterozygote that 100% for sure is heterozygous at the time was worth about $1,000. Okay, not as much as this albino, but definitely more than a $75 boring brown snake. But he's saying it's a 66% chance. If it's an F1 of a monohybrid cross, that's 100%. All of those are heterozygous. So what he did, he didn't have an albino snake. He crossed 2 albinos. And look at the 4 possible outcomes of crossing 2 heterozygous snakes. One of those 4 possible outcomes is albino. Is the snake he's selling for $450 albino? No. He knows it's not that. And so for that reason, that the snake that you're paying $450 for is one of these 3 here. It's either homozygous dominant and should really only be worth $75, or it's a heterozygote. Okay, so 2 out of 3 of the remaining possibilities for what this snake is when it was born are heterozygotes. Now, as I said, heterozygotes are worth over $1,000. Okay, so you're kind of paying somewhere in the middle. You're paying $450 for a snake that has a 2/3 chance, 2 out of 3 here, genotypes that are produced by his cross of a heterozygote with a heterozygote. You got a 2 out of 3 chance of getting an individual that carries that allele. So this is an interesting real-life example of how you can use the understanding of inheritance of certain alleles to calculate probabilities. And it's similar calculations that genetic counselors use today. They have a much better understanding of patterns of inheritance and the alleles that are present, and there's a lot more to it, but this is the basis of it, right? So now let's move on from this monohybrid cross example and acknowledge that there are much more complex crosses that take place. Okay, when you're crossing different plants, when Mendel's crossing plants, he didn't necessarily just cross plants that had purple flowers with white flowers. Sometimes he crossed a pea plant that had yellow peas that were round to a plant that had green peas that were wrinkled. Okay, so you can cross organisms when you breed puppies, when we breed cattle, when you breed anything. When you were born, it was a multi-hybrid cross, right? Your parents have very different genotypes, and they each gave you 1 copy of every gene they have. And it's all of those combinations that create the unique phenotype that is you, okay? Now, if we take Mendel's simple monohybrid cross and extend it to include another trait, crossing plants that differ in two traits, in this case, seed color and shape, the Punnett square gets a lot more complicated. Okay, so a monohybrid cross Punnett square is 2 by 2, but then when we add another character, it jumps to 4 by 4, and you add another character and it increases quite dramatically. Okay, and I want you to think about why this is. Look at, on the left here, we have a homozygous little r, homozygous big Y peat plant in the parental generation that's crossed with a homozygous big R, homozygous little y. So a round green plant crossed with a wrinkled yellow plant. And those F1 offspring, of course, have the heterozygous character or genotype for both shape and color. They have a big R and a little r. They have a big Y and a little y. Those are the F1s. They are all round. They are all yellow, 100%. Again, the F1s are quite simple. But when you cross an F1 with another F1, here's where it gets complicated. And here is why the Punnett square increases in size so dramatically. Okay, so look at the top of the columns and the left side of all the rows. Okay, what you see is that the first column is the big R with a little y. The second column is the big R with a little y. Third column, little r, little y, then little r, big Y. Okay? It's every possible combination that both the father and the mother could put into their gametes. Think about one of these F1 parents that is a pea plant that has round and yellow peas, but is heterozygous for both of those conditions. So into their gametes, they will put an R and a Y. But every combination is possible. Big R, big Y, big R, little y, little r, little y, little r, big Y, right? And each of those combinations is equally probable. And for that reason, in each row and column, there's a 1/4, okay, beneath the allelic combination that that parent has put into that particular gamete. So 1/4 of the gametes from each parent will have a big R and a big Y, okay? And so when you combine the rows and columns, you get the genotypes that are listed in each of these cells. And if you look at those genotypes, then the phenotype that's illustrated there will make sense to you. Now within each of those cells, you'll see there's a fraction, 1/16. Each of those cells is 1 out of 16, right? It's a 4 by 4 matrix. 4 times 4 is 16. There are 16 possible combinations here, but realize that not all of them are different from one another, okay? So look at, so not the very top left square, but look just below it. Big R, big Y, little y. Then look up 1 over 1. There's another big R, big Y, little y. There are 2 ways that offspring can be produced that are homozygous dominant for R and heterozygous for Y, okay? And in these, they are getting a different Y from the parents, the different parents, okay? They end up heterozygous for Y each way, but they're getting the dominant Y from a different parent, okay? So in this, you will see that there are lots of offspring that have the dominant phenotype. Remember, dominant is yellow and smooth. So look at how many yellow and smooth offspring there are, or possible combinations of alleles there are in this grid of 16. So the whole top row and the whole left column, right? So that's 4, 5, 6, 7. And then there are 2 more, 9. 9 of these 16 are round and yellow. You can see that there are 3 that are smooth and green, so they have the dominant shape, but the recessive color. There are 3 that are wrinkled and yellow. They have the recessive shape, but the dominant color. And only one out of these 16 combinations that are possible is wrinkled and green, recessive, homozygous recessive for both, okay? So exhibiting both recessive traits. So only 1 out of 16 is homozygous dominant for both. Only 1 out of 16 is homozygous recessive for both. But all those heterozygotes in between exhibit at least one, if not both, of the dominant traits. And so just as in the monohybrid cross of the F2 generation, we saw that the genotypic ratio was 1 to 2 to 1, and the phenotypic ratio was 3 to 1. In a dihybrid cross, we see a 9 to 3 to 3 to 1 ratio of phenotypes, okay? The genotypic ratio is a much longer string of numbers that we're not going to worry about, but I just want you to understand how a dihybrid cross works, and that the frequency of those dominant phenotypes is more common than the recessive, and that each of these possible combinations is equally probable and likely, and their frequency is of each of these phenotypes is dependent upon that. Okay. Now, it might strike you that this would be a very difficult question to answer if I gave you 2 individual parental genotypes for 2 genes and asked you what is the predicted frequency of individuals that are heterozygous for gene R and homozygous recessive for Y. Okay. Now, that might be a little difficult, and it's especially difficult if it's not a simple F2 cross. Okay. There is a mechanism that you can use to use your understanding of these combinations of alleles and their relative probabilities to calculate the frequency of any genotype and all genotypes from a dihybrid cross or even greater. I encourage you to watch this YouTube video that has horrible music accompanying it to understand how you can use what is called the forked line method. Okay? And the forked line method is a way in which you can calculate genotype frequencies, okay, for a cross. And that is an important skill. All right, so I want to talk now particularly about Mendel's 4th postulate, which is independent assortment. Okay, so the independent assortment of alleles is critical to the combined genotype of an organism, your genotype, all of the traits that you received from your parents, all the gene copies, all the alleles you received, those combinations that were put into the gamete by each of your parents are determined through this process of independent assortment. Okay, so traits are inherited independently, and what that means is just because you have green eyes like your mother doesn't mean you necessarily have let's say wisdom teeth like your mother does. There could be 2 traits that are genetically controlled and you get one of them from your mother but not the other. Okay, you might have the phenotype of your father for something else. And so the independent assortment is a very important concept for you to understand, and it stems entirely from the process of meiosis, and particularly metaphase 1. Okay, and the principle of the product law of probability applies here and helps us understand the expected frequency of different genotypes for multigenic crosses. And so independent assortment is simply the assortment of maternal and paternal chromosomes during meiosis 1. Okay, so you can see here that if we color code the chromosomes that each of these cells received from mother and father as blue and red respectively, so let's say blue comes from dad and red comes from mother, there are 2 ways that those homologous chromosomes can line up side by side in metaphase 1. So on the left, you can see that all of father's chromosomes that were given to this cell are on the left, and all of mother's are on the right. Whereas in the second possibility on the right, the blue big chromosome and the red small chromosome are on the left, and vice versa. What that means is that the gametes that are produced under possibility 1, you're going to get both of mom's chromosomes or both of dad's in the gamete. The second possibility, you can see that you get one of mom's and one of dad's in each of those gametes. And so this is a very different scenario. All right. So what is happening here is that chromosome 1, the big one, is lining up side by side independently. That's the independent assortment. They're lining up side by side independently of chromosome 2, the smaller one. It doesn't matter which one, whether the blue one's on the left or the blue one's on the right. Okay? The fact that the blue large chromosome is on the left has no bearing, no effect on the orientation of the smaller blue chromosome below, whether it's on the left, possibility 1, or it's on the right, possibility 2. Chromosome 1 and chromosome 2 here are independent of one another, and they assort independently. They don't affect one another. Okay? This process leads to this exceptionally high diversity that we observe in the possible gametes that can be produced. Okay? So the possible number of unique gametes that can be produced is 2 to the n, where n is the haploid chromosome number. Like, how many chromosomes do you have? That's 23. So you can produce 2 to the 23rd unique gametes. That's more than 8 million unique gametes. And your partner can produce more than 8 million unique gametes. Okay? And then there are 8 by 8 million unique combinations that can be created. Okay? You could populate an entire country of millions of individuals that are not identical. Okay? All right. So now I want to move on to one more topic that I want you to be familiar with, and we'll explore this a bit more in some of the lab work that we're doing. But this is, how do we use our understanding of these probabilities and use mathematics and statistics to determine whether certain traits are following the independent assortment and Mendelian patterns that we expect based on our understanding of genetics. Okay? And to do this, we use what is called a chi-square test. Okay? And a chi-square test is used, as I say here, to determine whether observed results deviate significantly from expectations. Okay? And you know what these expectations are now. Our expectations from a monohybrid cross are we expect 3/4 of the offspring, the F2 offspring, to exhibit the dominant phenotype, and 1/4 of the offspring to exhibit the homozygous recessive phenotype. Okay? So let's say we have 1,000 offspring. If of those 1,000 offspring, 740 have the dominant phenotype and 260 have the recessive phenotype, well, that's not exactly what I expected. At what point do we draw the line? How frequently, given the random nature of gametic fusion, might we observe a situation where 740 versus 260 instead of 750 versus 250? It's not a great departure, but again, a chi-square is used to help us say, yeah, there's, if it really is behaving like a Mendelian monohybrid cross, there's lots of wiggle room around that 75-25 split, so that it's reasonable that we might see a 740-260 split. Okay? But what if that number were 700-300 or 650-350? Okay? At what point do you draw the line? Well, we use the chi-square test to draw that line at around an area of possible outcomes that we would expect to occur 95 out of 100 times. Okay? You know, it's certainly possible that even if it is behaving like a Mendelian trait, you know, if we did this cross 1 trillion times, one of those times it might come out 250 dominant and 750 recessive, just by chance. It just could happen. It's not likely, but most of the time it's going to be pretty close. If it is a monohybrid cross of a Mendelian trait, it's going to be close to the 750, 250. So how do we use chi-square to see if this falls within what we expect 95% of the time in a Mendelian monohybrid cross? Well, the first thing we have to do is figure out how greatly our observed numbers deviate from expectation. And in this case, both of those deviations are 10. We've got 10 fewer dominants, we've got 10 more recessives. Okay? So 740 and 750, that difference is 10. 260 and 250, that difference is 10. Now, one of these we've got 10 less, one of these we've got 10 more. You'll see why that doesn't matter in a moment. Okay? So the chi-square calculation is actually the sum of these observed minus expected values. O and E refer to observed and expected. Okay? So 740 minus 750, okay, is 10. It's negative 10, but we square it. Okay, you see that? Observed minus expected. And it's divided by the expected number. And we do that with the 260, 250 number, also 10. So what we would end up with in calculating this chi-square number is 10 squared over 750 plus 10 squared over 250, right? The two different expected numbers, okay? And that sums to 0.53. All right, so that is how you calculate a chi-square. And there are loads of examples online if you want to work through more, and I'm certain there are some informative YouTube videos if you care to look for more chi-square calculation examples. But it's a pretty straightforward calculation. Now, what's not quite as straightforward is how you interpret that 0.53. What does that mean? Okay, there are a couple things that we need to do. We need to determine what's called the degrees of freedom, and I won't get into this too much, but basically we have two categories of numbers. We have the numbers of the dominants, the numbers of the recessive phenotypes. So we have two different classes. When we did the calculation above, we added two different things together. So that number of categories is 2. Degrees of freedom is this number minus 1. Okay? So for this, it's 2. If the number, instead of there being 2 categories, if there were 10, our degrees of freedom would be 9. Okay? So we are going to use, in the interpretation of our chi-square value of 0.53, we are going to use this chi-square table below. And these tables are used quite commonly. Hopefully you will take a statistics class and learn how to use these tables. Okay? I strongly encourage you to take a statistics class. They're incredibly helpful. All right, so the degrees of freedom is this left column, denoted by this script V. Okay, so our degrees of freedom is 1. So we're going to only be looking at this row number 1. Okay, and there are 5 different columns here, and it's always a bit confusing when you're first learning statistics. But basically, we are going to focus on the 0.05 column. It's the third column overall in this entire table. So the value we're looking at is 3.841. And what we're doing is we are comparing our number 0.53 to this. Well, 0.53 is much smaller than that. Let's think about what that means. Okay, the first thing we're going to think about is what the hell is 0.5? Okay, well, what percent is that? That's a 5%. Okay, 0.05 is 5%. Now, what we are using this table for is if your degrees of freedom are 1 and your number is greater than 3.841, okay, what that means is that your chi-square value is big. It's bigger than 3.841. It is a larger chi-square value than you would expect 95% of the time. Okay? Only 5% of degrees of freedom 1 chi-square values under normal circumstances, only 5% of the time would it exceed this chi-square value. Okay? This is a difficult concept to get, but let's think about it given these points here for a minute. We use this calculated value and we compare it to the table, our 0.53. Our null hypothesis, our expectation going into this is that the ratios we're seeing are what we would predict using Mendelian ratios. We're basically assuming, hey, these numbers, these 740 and 260, this is a Mendelian cross, okay? And this is what I would expect. It's a little off, but it's okay. I want you to think about how we got that 0.53. What would make that number larger? What would make that number larger than 3.841? What number in those previous calculations do we need to increase? If those numerators were greater than 100, okay, how do you get those numbers greater than, how do you get a chi-square value that's greater than 1 or greater than 2? Each of those fractions that we used has to be greater than 1, right? They'd have to be pretty big numerators over those denominators. That means there's a huge difference between observed and expected, okay? So if our number is smaller than 3.841, we're okay. Our null hypothesis is good. We are observing ratios that are consistent with Mendelian crosses. But if we got a chi-square value that was very high, higher than 3.841, we would say no, that null hypothesis does not fit this data. These data are so different from our expectation given Mendelian ratios that we would expect, that I would not expect to see this 95% of the time if it truly were a Mendelian cross. Okay, so this is a complicated concept to get. I think it would behoove you to read this bit of text in detail, maybe watch or read some additional material, and I'd work through problems number 15 and 16 in the text. And if you'd like me to send these problems to you, if you don't have them with you this week just yet, I can, I'll probably post an image of these on Blackboard, okay? All right, the last little bit of this chapter deals with pedigrees. And pedigrees are a way of illustrating families that have produced offspring, not always just 2 generations, it can be many generations, but it illustrates the gender or the sex, the biological sex of these individuals. You can see circles are female, squares are male, diamonds are sex unknown. We don't use those all that often. Individuals in which they do not have the trait are clear, so white circles, white squares. Individuals that have the trait that is being considered are filled. In the text, they're filled in blue. Examples you might find online are filled with black, okay? And then these individuals that are connected by these horizontal lines that go down to them, those are offspring, okay? So it'll make sense when we look at a pedigree here on the next screen. So here's a pedigree for the inheritance of an autosomal recessive trait. So autosomal, you may remember, that is a gene or a trait whose gene that controls that the phenotype of that trait is on one of your 22 chromosomes, not your 23rd sex determining chromosome. Okay? So let's look at this pedigree for an autosomal recessive trait. Now, in row 1, we have generation 1, and we have two different parental sets. Parent 1 and 2 and parent 3 and 4, and each of them has some children. Parents 1 and 2 have 3 kids. Parents 3 and 4 have 4 offspring. Okay? Now, 3 and 4, all we know is that those parents do not exhibit the trait. Okay? It's a recessive trait, so do we know their genotype? We don't know their genotype, but we know their phenotype is the dominant phenotype. Now, parents 1 and 2, parent 1 exhibits the trait. We know it's a recessive trait, so it is that individual is homozygous recessive. Okay? Parent number 2 there does not exhibit the trait, has the dominant phenotype, and therefore could be heterozygous or could be homozygous dominant. All of their offspring do not exhibit the trait. So given that parent 1 is always giving each of their offspring that recessive allele, it seems like parent 2 might not be heterozygous. Okay? There were 3 opportunities they had. Each of those offspring of theirs had a 50% chance of getting the recessive allele. If parent 2 had it, then they didn't. Okay? Now, parents 3 and 4, they had 4 offspring. None of them exhibit the recessive trait. They are all exhibiting the dominant trait, but we don't know their genotype. Now let's look at the 3rd generation. Here we see in the 3rd generation, offspring 3 and 4 exhibit this recessive trait. So what's their genotype? They have to be what? They're homozygous recessive. How did they get to be homozygous recessive? They had to have received the recessive allele, one from each parent. So it doesn't surprise us that in the 2nd generation, parent 3, that square, had the recessive allele. Their parent, number 1, was homozygous recessive and had to give it to them. So we know they're heterozygous. Okay? But if this 3rd generation blue square, that individual number 4, is homozygous recessive, it got a recessive allele from its parent number 3, but it also had to get one from its parent number 4. So in looking at this, you can tell me in generation 2, you know the genotype of individual 1, 2, and 3, and 4, for sure. Okay? You know that individual 1, 2, and 3, and 4 all have to be heterozygous. Okay? They are all heterozygous, every single one of those, 1, 2, 3, and 4. Okay? Think about it. They all had to get, 1, 2, and 3 had to get the recessive allele from their father, number 1. Okay? And they definitely didn't get a recessive allele from their mother, number 2, so they have to be heterozygous, right? Individual number 4 in that 2nd generation, the mother of this 3rd generation, she has to have the recessive allele so that she could give it to her offspring, number 3 and 4, that are both homozygous recessive. Of course, those 2 got the recessive allele from their father, okay? But they also had to get it from their mother. So we know that that mother is heterozygous. Could she be homozygous recessive? No, because she would be blue, all right? Her circle would be blue. So since we know that she's heterozygous, what do we know about her parents, 3 and 4, in the 1st generation? They absolutely, one of them had to be heterozygous, okay? Now, could both of them have been heterozygous? Sure, it seems less likely, but it's possible, okay? So, what about in the 3rd generation? Individuals 1 and 2. I want you to think about them for a minute. Can you tell me their genotype for sure? That's the type of question I want you to be able to understand and tell me and explain, okay? If you need to know whether you can or cannot infer their genotypes, okay? Let's go to another situation in which there's an autosomal dominant trait, unlike the previous one that was recessive. Okay. Now, the one thing you'll notice in the dominant trait is that unlike the recessive trait, that recessive trait completely skipped the second generation. Remember, there were no blue circles or squares in that second generation. That is impossible for a dominant trait. A dominant trait can't persist to the third generation and not have showed up in the second, okay? Because it's dominant. It has to show up. If an individual has it, they have it, and they express it, and they show it in their phenotype. So here we have an autosomal dominant trait that is present in the male and not in the female. Do we know if that male is homozygous or heterozygous? Not just by seeing his phenotype, but when we look and see that he has offspring, like his offspring number 3 and number 4, that do not exhibit the trait. Now do we know his genotype? We sure do. He had to have been able to give them the recessive allele because they do not have a dominant allele. So he's heterozygous. So parent 1 in the first generation is heterozygous. Parent 2 has to be homozygous recessive because they're not blue, okay? Second generation, individual 1, definitely heterozygous, right? Had to get the recessive from the mother, number 2. The partner in the second generation, number 2, doesn't have the trait, homozygous recessive. Every individual in this pedigree, that circle or square, that is not blue, is what genotype? They're all of them homozygous recessive, okay? So, because of this, look at this pedigree. Are there any of those blue individuals that are homozygous dominant? You should be able to answer that too, okay? Now, the text on page 52 also has a few problems that would be good pedigree practice as well. So I encourage you to review those. And again, I'll post those as well on Blackboard. Okay, so this is where we end chapter 3, the inheritance of Mendelian traits. The next chapter deals with traits that are inherited in a non-Mendelian fashion, so they're not inherited in quite as simple a way. But with this foundation that you have from chapter 3, you should have little to no trouble with chapter 4. Okay.